Alcoholism, as defined by the Webster Dictionary, is the continued excessive or compulsive use of alcoholic drinks. However, could it be that this addiction is more than a harmless habit turned horrific, more than an innocent desire turned captive? Around 40 to 50% of the risk of alcoholism is caused by the environment and the activity between the environment and one's genes. The remaining 50 to 60%, however, stems from deep within, tracing back to one's genetic material. But before we dive into the biological factors involved in alcoholism, let's take a look at the presence of alcoholism in society today. In a study published by JAMA Psychiatry, researchers found that 1 in 8 American adults, or 12.7% of the U.S. population, meet criteria for alcohol disorder. All of these people, victims of alcoholism, have one thing in common: the certain functioning of certain genes. While there are many variations in specific genes that are associated with alcoholism, including those that control the breakdown of alcohol in the body, 2 in specific are the OPRM1 gene and the SLC6A3 gene. The OPRM1 gene provides instructions for the coding of the protein mu-opioid receptor. According to Genetics Home References, the opioid receptor are a part of the endogenous opioid system, which is the body's internal system for regulating pain, reward, and addictive behaviors. These receptors are found in the outer membrane of nerve cells found throughout the nervous system and binds beta-endorphin. Consuming alcohol leads to the enhanced release of these beta-endorphins, reducing pain throughout the body. The SLC6A3 gene provides the instructions for the creation of the protein dopamine transporter. This protein is found in the membrane of neurons in the brain and has the job of transporting dopamine, the chemical messenger that activates the reward center of the brain, into the cell. When an individual stops drinking alcohol, dopamine levels in the body are reduced, thus reducing the buzz caused by drinking alcohol.
We plan to include the best 12 studied genetic variations that affect alcoholism on our SNP test for SNPs associated with alcohol dependence. The 2 SNPs that we believe are the best predictors of risk for alcoholism and will most definitely be included in our test are the RS1799971 and the RS27072. The RS1799971 is a single nucleotide polymorphism found on the OMPR1 gene, mentioned earlier, that influences the intensity of alcohol cravings. The possible genotypes for this gene include being homozygous for the A allele, being homozygous for the G allele, or being heterozygous with one of each allele. Scientists have found that those with the G allele bind the beta endorphins 3 times as tightly than those with the A allele. This means that the digestion of alcohol suppresses pain more efficiently in the people with the G allele, leading to a craving of the body for this kind of pain relief. Those with at least one G allele experience around 2.5 times stronger alcohol cravings than an individual who is homozygous for the A allele. The RS27072 is a single nucleotide polymorphism in the SLC6A3 gene, also mentioned earlier, which holds the instructions for producing the dopamine transporter protein. The possible genotypes for this gene include being homozygous for the C allele, being homozygous for the T allele, or being heterozygous with one of each allele. Those homozygous for the C allele have 2 times the risk of experiencing severe withdrawal symptoms. This SNP decreases the amount of dopamine transported when alcohol is not ingested and is associated with severe symptoms of alcohol withdrawal, the most severe symptom being seizures. This can lead to a higher risk of the development of an alcohol dependence.
Using our test can help an individual determine whether they should take precaution around alcohol. Alcoholism is not necessarily a life-threatening condition, but anyone who wants to be aware of genetic proneness to alcoholism should take the test. Patients with the RS1799971 nucleotide polymorphism that we will test, found to have the G allele, will have a 250% higher craving for alcohol. If people know about how their body is programmed to act on alcohol, they can guide themselves on how to correctly act around alcohol. Our test can also help alcoholics determine whether their addiction is catalyzed by the presence of alcoholism-related alleles or a sole product of unhealthy environmental effects. Even though our tests look at the best 12 studied genetic variations that can affect alcoholism, we cannot test for all possible genetic influences that can affect an individual's risk. Even though an individual can be tested positive for a high risk of alcoholism, they may go on to live an alcoholism-free life. Vice versa, if an individual tests negative for a higher risk of alcoholism, we cannot confirm 100% that the individual will not develop alcoholism later in life because we have no way of testing environmental factors. Whether the genes tested are present or not, any human still has a minimal risk of becoming an alcoholic, and that is a very important thing to consider when talking about the topic of alcoholism.
Testing for traits that can cause alcoholism can be an emotional and financial burden. Knowing that you are alcoholism prone may cause you to be more cautious, even to the point of being fearful around alcohol. Financially, testing can be expensive, though it may help save you money on alcohol, and the expense is well worth not becoming an alcoholic. Another ethical consideration is that people could be labeled an alcoholic without actually being one, just because of their genetic makeup. All of these are possible concerns in genetic testing for genes related to alcoholism.